All right. So now that we've built our base of how the nervous system is organized and what kinds of cells make it up, let's now talk about how it works. So you've probably heard of the term action potential and correlate that or have some association with that being how neurons send their signals. But before we can talk about action potentials, we need to talk about this word potential, what that means, and how all that gets manipulated through things like graded potentials and what effect that has, etc. So at rest, your neurons and really all of your cells have what we refer to as the resting potential. And this is describing the charge, so potential is referring to charge, of the membrane of that cell. So at rest, what is the charge of the membrane of these neurons? So this is a non-signaling, not active, unstimulated neuron. It is at rest. No signal is being sent. And at rest, due to the concentration of various ions and proteins and some larger molecules, as we've discussed earlier in this chapter, or not in this chapter, earlier in this course, right? You have different concentrations of these chemicals and ions on either side of your membrane. That gives the inside of our cell membranes a relative charge compared to the outside. And we are concerned about what charge is on the inside of that cell membrane. So you can see this is the cytosol on the bottom here. And relative to the ion concentration outside of this cell, the internal portion of the membrane has a negative 70 millivolt charge. So you can see that being measured here. It's measuring the charge of the inside relative to the outside. And at rest, when it is not sending a signal, this is at negative 70 millivolts. And this is constantly being upkept by a few different systems. So first off, this is where we're mainly going to be applying this idea of major extracellular and intracellular ions. So inside the cell, our major ions or charge or things that are contributing to the potential essentially are potassium. So these K+ ions, potassium is in higher concentrations inside the cell. We have phosphate groups and then various proteins that are contributing to our internal charge here. Outside of the cell in the extracellular fluid, we have much higher concentrations of these positively charged ions and chemicals compared to inside. So we have high concentrations of sodium, high concentrations of calcium, it's not included on this list here, but then high concentrations of our anion chloride. So since the internal side of this membrane has a negative charge compared to the outside, and a relatively strong negative charge, negative 70 millivolts is not small, that is going to cause an electrical gradient, right? Opposites attract in chemistry. So anything with a positive charge that's outside the cell is going to electrically be drawn to move in to the cell. And so vice versa, negatively charged ions inside the cell are going to be drawn out of the cell. And positively charged ions are going to be drawn to stay there and not be pushed out. But electrochemical charge or electrical charge isn't the only thing we have to consider here. So even though we have this idea of opposites attract, negatives pulling positives, positives pulling negatives, we also need to consider concentration. Because that is going to drive or cause the movement of these molecules or add to the kinetic energy that is going to drive the movement of these molecules. And so we have to combine those two ideas of knowing what the concentration of different ions is in these various spaces and what the charge is. And the combination of those two forces is what is known as the electrochemical gradient for these ions, and that is going to drive a lot of their movement. I guess I'm kind of getting ahead of myself, but we're going to come back to that later. Electrochemical gradient. It's on the next slide or the next two slides. That's our big driving force, which is the combination of the electrical gradient and concentration of these chemicals. But at resting potential, we have a few different systems in place that are keeping that potential the same. So we have a few of what are called these ion leak channels. So we have a calcium leak channel, we have a sodium leak channel, And those are just channels that are constantly open that allow for small amounts of these ions to consistently diffuse out of the cell for potassium or into the cell for sodium. And then we have a protein pump. So this is going to be active transport, whereas those we're allowing for passive transport, called the sodium-potassium exchange pump, which is then going to cancel out that movement, essentially, which just ends up providing this balance that keeps the membrane potential at negative 70 millivolts. Okay, so the reason we have essentially potassium leaving the cell and sodium entering the cell with these leak channels open, and the reason that the number of these things is different also, right? We see that we only have 2 potassium leaving, whereas we have 3 sodium entering, comes back to that electrochemical gradient. Okay. So this can be an overwhelming slide, so trying to go one at a time. Essentially, what we're looking at here is the combination of these 2 forces, our electrical gradient and our chemical gradient, which is the concentration. The overall net charge of our electrochemical gradient for both of these ions. And then what would happen to the membrane potential if the membrane was freely diffusible, right? Our cells have selectively permeable membranes, which is why we need things like leak channels and pumps to control the movement of these ions. But if they were freely permeable, we'd end up with very different charges. So looking at the electrochemical gradient for potassium, potassium is in much higher concentrations inside the cell compared to outside. So the chemical gradient, the concentration gradient, is strongly trying to drive potassium out. It's a high concentration in, a low concentration out, so those potassium ions want to get out. However, the electrical charge outside of the cell is positive compared to the inside of the cell. So electrically, they're actually being drawn back into the cell. But that magnetic electrical gradient is going to have an overall weaker force compared to the chemical gradient because of how strong the differences in concentration are. And so the overall net electrochemical gradient on potassium is pushing it out. So we have a relatively weak electrochemical gradient pushing potassium out of the cell. And so when these leak channels are open and there is space for potassium to move, it is naturally going to be moving out of the cell because of that combined electrochemical gradient. For sodium, we have a chemical gradient pushing them into the cell. So we have the opposite concentration gradient. We have a high concentration outside of the cell for sodium and a low concentration inside. So they strongly want to be inside from that chemical gradient, and the electrical gradient is moving in the same direction. Right? Sodium is a cation, it's positively charged. The inside of the cell has a slightly negative charge compared to the outside, so they are electrically being pulled into the cell as well. And so if they can move across the membrane, there's going to be a strong net charge pulling them in. So potassium had a relatively weak net charge sending them out. Sodium has a strong net charge pulling them in. And so when both are allowed to enter or leave, we get a smaller amount of potassium leaving compared to the amount of sodium entering. 3 sodium ions come in at a time, 2 potassium ions leave at a time. And then if we have access to energy, the sodium potassium exchange pump will basically cancel that out. It'll bring 2 potassium in for every 3 sodium it kicks out. And so this is really the main idea here, because besides these leak channels, we're going to have other channels that open and provide even more space for some of these ions to move at different times. And so when they can cross the membrane basically freely, potassium is going to leave at a medium rate, and sodium is going to enter at a fast rate. And so the bottom parts here are showing what would happen to the overall charge of our membrane if there was no selective permeability, if these ions could just freely cross. And overall, again, that gradient for sodium is going to be stronger than the one for potassium. So if these could just freely move, some more potassium would leave, which is going to take that positive charge with them, so leaving the inside of the cell slightly more negative at negative 90. But if sodium can freely enter, it is going to very significantly change the charge of our membrane, driving it all the way up to around plus 66 millivolts. And so these two changes in charge are essentially what are going to happen if we open and close different ion channels. So if all the sodium ion channels are closed and all the potassium ion channels are open, our membrane potential is going to get more negative. If all the potassium channels are closed and all the sodium channels are open, the membrane potential is going to be strongly positive. And so it is this constant manipulation and change between charge of the membrane that conducts an action potential or conducts a signal along the membranes of these cells. So that is all facilitated through a few different types of these ion channels. So we already talked about the leak channels. We introduced the sodium-potassium exchange pump. So those things are basically just always open. The leak channels are not really modulated or regulated. They're just constantly open. The sodium-potassium pump is constantly pumping as long as it has access to energy. And your neurons are very energy hungry in order to keep this system going. But what is going to cause these larger changes in action potential, or sorry, in membrane potential, are coming from chemically regulated channels, voltage regulated channels, and mechanically regulated channels. So a majority of the signal starting for our neurons is going to come from chemically regulated channels. So some sort of molecule, like a neurotransmitter or a hormone, binds with this protein channel, which causes it to change shape and open up, which now allows for the diffusion of more ions. So they are chemically regulated. They're being regulated. Their opening and closing is regulated by some chemical, a ligand, that binds with it and opens or closes that channel. So this is what is going to be doing a majority of the signal starting that allows for large amounts of these ions to start moving in or out of a cell. The effect of that ion movement is then going to change the membrane potential, which could open or close different voltage regulated channels. So here we see an example of our sodium voltage regulated channels. At resting potential, the activation gate is closed, so sodium can't go through that channel. But if we increase the membrane potential a little bit, make it a slightly more positive to threshold, which is negative 60 millivolts, then those those voltage gated sodium channels open, and now sodium can flood in at much higher amounts, which is going to significantly increase the charge of the membrane until the opposite side finally closes at the next threshold, which is plus 30 millivolts. So various charges, various voltages have different effects on those protein channels. Right? Different sides can open or close, etc., as that membrane potential changes. And then lastly, our least common of our control mechanisms here for these ion channels are going to be mechanically regulated channels. So based on the physical movement of the membrane, that is going to cause separation or reattachment of the proteins that make this channel. And so if some pressure is applied to this membrane here, you can see that those proteins have now separated, and now this channel is open, which allows for things like our sensation of pressure and touch. But mainly, we are focused on these two as far as understanding potential and how neurons signal each other. So, action potentials begin at the axon hillock. And whether or not an action potential starts is determined by graded potentials. So graded because they can be excitatory or inhibitory. They can be positive or negative. There is a scale, a grade, right? There could be any number in between. And so the combination of input, the combination of stimulus is going to determine whether or not we have reached the threshold to start an action potential. So graded potentials start action potential. And they do this via depolarization. So I suppose that's an idea we should get this term. We should spend some time on this term for a second before I move on. So if at resting potential, we have this significant difference in charge, right? We have a negative charge inside and a positive charge outside. You can consider that a polar structure. Just like with polar molecules, where there is a positively charged and a negatively charged side to the molecule, our membranes are polar because we have a negative charge on one side and a positive charge on the other. And so we can depolarize that membrane by bringing this number closer to 0, or we can hyperpolarize this membrane by taking that number farther from 0. So decreasing or increasing the difference in potential. So depolarizing is decreasing that difference, so going from negative 70 millivolts closer to 0. Hyperpolarizing would be taking that number farther from 0, so going from negative 70 to like negative 90. So graded potentials, if they depolarize the membrane to what is considered threshold, which is negative 60 millivolts, then that will start an action potential. If they do not reach that negative 60 millivolt threshold, no action potential starts. And so that is going to come from a combination of signals. Some signals are going to depolarize the membrane. Some signals are going to hyperpolarize the membrane. And so if depolarizing signals are stronger than the hyperpolarizing ones, then you get an action potential. If not, they cancel each other out. So anyways, at resting state, none of our chemically gated ion channels are open. We just have our leak channels. Some signal, such as a chemical, a ligand, like acetylcholine, can bind with those chemically gated ion channels, opening them up, which is going to allow for the free movement of typically sodium. Typically, we're looking at sodium-gated channels for the start of a potential. So that's going to allow for sodium to flood in at a high rate. Remember, there's a strong electrochemical gradient pulling sodium into the neuron. And those sodium ions are then going to spread out, which is going to start changing the membrane potential of the membrane around that area. Right? So you can see as these ions came in, they don't stay in one area. They now want to diffuse away into all this open space that they have available. So they will start spreading down the membrane. And essentially, if this negative 60 millivolt charge reaches the end of the axon hillock, the initial segment of our axon, that is going to start a domino effect of voltage-gated ion channels for sodium opening and opening and opening. So we get more and more sodium flooding in as it moves down the axon. So that's the idea. Graded potentials are going to be a combination of depolarization and hyperpolarization. And if we have enough depolarization to reach negative 60, an action potential will begin. So action potentials begin at that initial segment, at the edge of our axon hillock. So at resting, again, both of our ion channel or gated ion channels are closed. We just have leak channels open. If the graded potential from the axon hillock allows for enough sodium to flood in to bring that negative 60 millivolt charge or change the membrane of the axon to negative 60 millivolts, that is going to open our sodium channels, our voltage-gated sodium channels. And so now a whole bunch of sodium can flood into the axon, which is going to cause rapid depolarization. So we're going to quickly jump from negative 60 all the way up to plus 30 millivolts. So that is depolarization during action potential. At plus 30 millivolts, those sodium channels are going to close, so no more sodium can come in to this part. And instead, potassium channels are going to open. And so now potassium is going to be flooding out at a much faster rate than it would have normally, because now we have a bunch of sodium that has entered too, changing the potential. So now we have an electrical gradient pulling potassium out too. So now potassium is going to leave the cell at a relatively fast rate until we get what is considered hyperpolarization. So all the way to negative 90 millivolts, where now those potassium channels are going to close. So both the sodium and the potassium channels have fully closed at negative 90 millivolts, and the sodium-potassium exchange pump and those leak channels are going to help get our membrane potential back to its resting state at negative 70, and then this process can start again. So again, at negative 70 millivolts, both of those voltage-gated channels are closed. At negative 60, sodium-gated channels, the sodium channels open. So sodium comes into the cell until plus 30. At plus 30, the sodium channel closes and the potassium channel opens. So now potassium is going to flood out of the cell until it reaches -90. And at -90, the potassium channel is going to close. So now they're both closed again, and the sodium potassium exchange pump will bring things back to normal, back to resting state. It's basically all described on this reference slide here. Okay. So there's a few other specific differences between graded potentials and action potentials besides just where they're occurring, right? So graded potentials depolarize to threshold, which is going to cause an action potential. Graded potentials happen at the axon hillock. Action potentials occur down the neuron. But some other important differences are that graded potentials could be either depolarizing or hyperpolarizing, right? We can have excitatory signals, which would be depolarizing, and inhibitory signals, which would be hyperpolarizing, right? So the combination of overall all these different signals that one neuron may be experiencing is going to determine whether an action potential starts. An action potential is always depolarizing. It is starting this signal and sending it down a bunch of those voltage-gated ion channels. And once an action potential has reached threshold, so if the axon gets its membrane potential to -60 millivolts, an action potential is going to begin, and it is going to travel down the entire axon to the synaptic terminal. So that is considered all or nothing. And if it reaches threshold, it is going to go the entire way. Graded potentials don't have that. So let's see, what else here? Graded potentials passively spread, and so they decrease in intensity the farther you get from the source of that graded potential. Whereas, again, action potentials are all or nothing. So the entire axon is going to experience the exact same changes in potential as that action potential travels. And then one of our biggest ones, most important, impactful differences here. Graded potentials have no refractory period, meaning that you can continuously stimulate the axon hillock of a neuron with depolarizing and hyperpolarizing signals. And it's just continuously happening, continuously happening, continuously happening. Action potentials, however, have a very short refractory period, meaning that for a short amount of time, it is impossible for another action potential to just start again. So an action potential goes down the axon. It's going to take a couple seconds there. I don't know the actual time, milliseconds, maybe even smaller. Okay? But there is a little bit of time where that axon needs to regulate itself, get itself back to resting state before another action potential can start. And that's basically because of this hyperpolarizing period, because so much potassium leaves while this voltage-gated channel is open, and these channels are slow to close. The voltage-gated sodium channels are pretty fast. Like once they reach their number, right, the -60 and plus 30, right, those two different sides open or close. But potassium-gated or the voltage-gated potassium channels are technically starting to close at -70 millivolts, but won't finish closing until -90. So you get that extra bit of potassium that is leaving. And so we have hyperpolarized this membrane. The charge is more polar than it needs to be. And so during that little bit of time, as the sodium potassium exchange pump is going to get this -90 millivolts back to -70, there is no way for another action potential to start. And that's that refractory period. So yeah, that's the difference between graded and action potentials. Graded potentials determine whether or not an action potential is going to start, basically. And so for most of our neurons, it's not as simple as just this empty membrane traveling down the entire axon. We have some help from oligodendrocytes or Schwann cells along that axon, which myelinate them. So again, myelin is referring to these fatty sheaths that cover different segments of the axon. And so each of these segments that are covered with myelin, those are considered internodes. And those are going to make up the space in between these tinier exposed segments called nodes. So the space between a node is an internode. Myelin fills the space between nodes. Those are internodes. And as I mentioned earlier, this myelin basically just keeps all of the ions that have entered the cell into the axon. So all of these sodium ions that are flooding in during depolarization are going to consistently diffuse down the axon, and they have no ability to leave until they reach the next node. So we get a consistent membrane potential from one node to the next, which just speeds up this process. We don't need to open more channels and consistently do that over and over again. Those channels just need to open and close again at the next node, and then we have more sodium diffusing to the next, etc. So this just allows for that signal to pass down the axon faster. That is referred to as saltatory propagation, right? Because we're moving salts, which are ions, propagating the movement of those salts down the axon, saltatory propagation. And so without myelin, that's considered contiguous propagation, which just takes longer because you have more of those channels that need to open and close all the way along the membrane of the axon, every little tiny part. Whereas myelin, for saltatory propagation, cuts out that middleman and allows this process to happen much faster. So you can see that happening here. So we have our action potential beginning at the initial segment of our axon. All this sodium that is flooded into the cell is going to start diffusing down, and it can't leave. It can't diffuse out, so it goes all the way to the next node where we have our next set of voltage-gated ion channels opening and more sodium flooding in. That sodium then goes to the next node, etc., and that just continues. So eventually we're going to reach the end of our axon and get to our synaptic knobs. And at the synaptic knobs, these action potentials are going to stimulate the release of some chemical. And so chemicals in our nervous system that send these signals are called neurotransmitters. And then those neurotransmitters are going to have some effect on the next cell, either acting as a graded potential on the next neuron or causing some response such as muscle contraction on a muscle cell at a neuromuscular junction. Since we've already discussed neuromuscular junctions, let's use this as our example, talk about what happens. So you should be familiar with the idea that an action potential reaches the end of the axon of this neuron that causes the release of acetylcholine, which is our neurotransmitter here. Acetylcholine binds with these chemically gated sodium channels, which allows them to open, and sodium rushes into the cell, which causes an action potential on that muscle. The parts of this we didn't really discuss, I guess, was the movement of that, or now you should have a better understanding of why that movement of sodium, why these these ion channels matter. So sodium rushing in depolarizes the membrane, which causes an action potential on the muscle cell. But something else that we didn't discuss is how these vesicles with acetylcholine end up being released, right? Stimulated to release, because it's not just the arrival of this signal, this action potential that does it. The action potential is going to cause the opening of different voltage-gated ion channels. Excuse me for a second. Sorry, I hit the wrong button there. I hit pause share instead of pause recording, so we'll see if I remove that sneeze. Anyways, the action potential reaches the synaptic terminal. That depolarization of the membrane is going to activate voltage-gated calcium channels in this case. So calcium floods into the synaptic terminal here, or sorry, synaptic knob. That influx of calcium is going to stimulate the exocytosis, essentially the fusing of these vesicles with the membrane. So exocytosis of acetylcholine. And then again, acetylcholine binds with these chemically gated sodium channels. Sodium rushes into the cell, which depolarizes the membrane and causes an action potential to propagate along the sarcolemma. So here. And so not all neurotransmitters are excitatory in the same way that acetylcholine is, and they don't all act directly in the way that acetylcholine did. So acetylcholine is quite simple. This is an example of a direct action of a neurotransmitter. So the neurotransmitter was released, it bound to some membrane protein, and that had a direct effect, such as sodium now flooding into the cell. The protein opened a channel and sodium flooded it. And this was not only direct, it was excitatory in the case of acetylcholine. But we can also have indirect effects, which means that neurotransmitter binds with a membrane protein. That causes the membrane protein to activate some other protein, which may even activate another protein. And then that protein goes and does something, such as a lot of the genetic control, so causing the expression of certain genes to produce these various proteins in response to neurotransmitters. So like a neurotransmitter could stimulate cell growth by acting as a growth factor, which now is then going to tell the nucleus of the cell to start expressing the genes related to cell replication. So that is an indirect effect and that can happen via G proteins or these other intracellular enzymes. But both of those are indirect because the actual neurotransmitter itself does not directly have its effect. There has to be these other middlemen like G proteins and cyclic AMP or these other enzymes and secondary messengers. So beyond mechanism of action, that could also be excitatory or inhibitory. So either making something happen or slowing something down. And so we can have these effects happening at either our axon hillock or graded potentials or at the end of the axon itself. So first off with graded potentials, like we said, these are going to be a combination of depolarizing and hyperpolarizing effects. So a combination of excitatory and inhibitory effects. And if the combination of those effects is strong enough to reach threshold, then great, you get an action potential. And so that combination could either come from temporal summation, or the combination of these things is referred to as summation, the sum of these signals. And that could either come from temporal summation, meaning the same signal being sent at different times, building up into a larger effect. So over time, the same signal coming multiple times could build up to causing depolarization to threshold. Or we can have spatial summation, where multiple axons are synapsing at that axon hillock. And so if they are both sending the same signal at the same time, that combination, or even if they have different signals, it's still spatial summation. It's the summation of all the signals in that space. So it could be temporal, meaning referring to time, or spatial, referring to how close two or more different axons are on the same neuron. And so if you have an excitatory postsynaptic potential with an inhibitory postsynaptic potential, together, those are going to cancel each other out, keeping them at resting potential. That's basically what's being shown here. EPSP is excitatory postsynaptic potential. IPSP is inhibitory postsynaptic potential. Postsynaptic referring to the synapse of the previous neuron. Anyways, excitatory signals and inhibitory signals are going to summate, combine, to determine the overall effect of that graded potential. And so our last concept here is we also have presynaptic regulation, meaning that some neurons are going to act directly, have their neurotransmitters being released directly onto the synaptic terminal of another neuron. So basically the two tails are meeting each other. Axons meet with other axons. And so you could have one neuron responsible for regulating the activity of the second one, and that is considered presynaptic regulation. So you could inhibit the release of neurotransmitters from that next neuron, or they could facilitate and stimulate the release of those neurotransmitters. So here we have a synaptic knob of a presynaptic regulator, and then the synaptic knob of the one being regulated, right? So this one is sending signals that are going to have an effect on the release of neurotransmitters from this other neuron. And so yeah, sorry, I guess I went a bit fast there. Where did my slide go? Just gonna back up. I guess there really isn't a whole lot to show here. Anyways, so in an inhibitory response here with a chemical like GABA, right, that is going to decrease how much neurotransmitter is being released. Thus, it doesn't matter if an action potential from this neuron has arrived, it has been inhibited, its action has been blocked, and so those neurotransmitters were not released. It has been inhibited. If it's facilitated and activated, it just essentially helps with more of that neurotransmitter being released. It doesn't necessarily cause neurotransmitter release on its own, but is going to facilitate more neurotransmitter release. And that brings us to the end of this slide deck. So, as always, if you have any questions or concerns, please reach out. And as I mentioned at the very beginning of this chapter, this is essentially the beginning of the end. We are going to use this as the basis to build on the rest of the chapters through Unit 3, which are all focused on the nervous system. I'll see you guys in the next one.